And the names for each of these, with your purines, it's going to end in SINE. So SINE for nucleoside. But with the pyrimidines, it's going to be like cytidine or deoxythymidine. The deoxy is something we're about to come to. And that means that you have one of these nitrogenous bases bound to this sugar here. There should be a hydroxide here. I'm sorry. And then what happens when you have deoxy? Well, in that case, you're going to just put a deoxy prefix to the front of that. So you have, say, deoxyguanosine. Deoxyguanosine. It sounds like the weirdest thing ever. Or deoxycytidine. Or deoxyadenosine. And what would thymine bound to a deoxyribose be called? Shout it out. Yeah. So when you have a thymine bound to a deoxyribose sugar here, it's a nucleoside. And what do you call it? What is the name for those two bound together? So here with cytosine, you have deoxycytidine. With thymine, it's the same family, it's a pyrimidine, so it has the same suffix. So it's what? You had it? Deoxythymidine. Exactly right. So all of these names, they seem really complicated and they get really long, but each little part just means one small thing. So that's saying deoxy, it's bound to a deoxyribose. Thi, which means it has a thymine in it, and dine, meaning it's that group bound to one of these sugars. Does that make sense to everyone? Am I like going too fast, or should I go back and cover something? No? Seriously, if something's confusing, raise your hand. Okay. Moving on, you can also have phosphate groups bound to this. I'm going to just erase this for right now and draw it all together. So you're going to have, say, adenine here again, and that'll be bound to your ribose. OH there, H, 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 H. And these hydrogens really, really like to bind to these carbon atoms because those are just the simplest bonds to form. And there's lots of free-floating hydrogen everywhere. So you'll have carbon with a hydrogen there. And normally it has a hydroxide there. But it likes to bind with the extra oxygen that was on the phosphate group. Remember it had oxygen there, oxygen there, oxygen there. And when an oxygen and a hydroxide combine, what do you think you get?
If oxygen or hydroxide, obviously?
Yeah. Am I doing that wrong?
No, I think you're doing it wrong. It should be a hydrogen and hydroxide.
Yeah. So there is a hydroxide here on the basic phosphate. So these will bind, and some of this will break off, and you'll actually get water from this junction. And then you end up with that phosphate coming down, and then you have it bound right there. So you have your phosphate bound to your nucleoside, which is a phosphate, a ribose, and a nitrogenous base, all three together. And these are called nucleotides. So you'll have nucleotide. You can think T for three parts. There's the phosphate, there's the sugar, there's the nitrogenous base.
That's really important.
Yeah.
Like they test over so much DNA vocabulary because you have nucleotides, nucleus, nucleosomes, nucleolus, all these like nucleo things that are kind of pretty different. So
Yeah.
you should make sure like
Um,
know how to see.
And the way, the way you'll actually name these is a lot simpler than the nucleoside, even though it's a more complex thing. So you'll say what base it has in there with that prefix. So, maybe it's not simpler. So you have the name of your nucleoside. So in this case, it would be what? Adenine plus a ribose is called adenosine. Right. So adenosine, and it has one phosphate here. It'll be called adenosine monophosphate.
What happens if it has 3 phosphates?
Well, if it has 3 phosphates, how do you think the name will change? Triphosphate, right. And in that case, I'm going to kind of draw over all this here. This oxygen will bind with another phosphate group, and you'll have P, O, double bonded O, another O minus. So this has 2. It would be adenosine diphosphate. Does that sound familiar? Anyone who's taken biology? Adenosine diphosphate.
ADP, adenosine triphosphate, ATP.
There you go.
Energy.
Okay. So these phosphates, because it has this negative oxygen ion here, it's really ionized, and so they hate each other. They do not want to bind with each other. It takes a lot of energy to bind them. So when you pack on even more energy, you're going to get another bond, and this binds to another phosphate. I'm not going to draw it here because I don't want to go over his stuff, but you're going to have
And then when you break those bonds, you release a lot of energy that's used to drive cellular processes like cellular respiration or photosynthesis, right? So that stuff happens.
yeah. So you'll get a structure that looks pretty similar to that. And this would actually bind to another nucleoside. So that's what I was about to cover here before I got off on that tangent. So this ADP will bind with another phosphate to make ATP. And again, those bonds that the phosphate groups make, because it takes so much energy to force them together, will have a lot of energy, and that drives most of the reactions within a cell. So these phosphates like to chain together with 2 riboses. So what will happen is you have, take this out so I have AMP again, adenosine monophosphate. This will bond with phosphate group, and you get another phosphate. And this oxygen will want to bind with the ribose, just another ribose that's down there, and it'll keep binding and make these really long chains, which we know as DNA and RNA. So this makes kind of like the backbone. At the bottom, it'll be finished off with an OH there. And at the top, this will have another ribose. And it has a hydroxide there. These hydroxide ends are important because they control the way it reacts. And when you have a small molecule that defines the way something reacts with another molecule, what's that called? We covered it last week. You have a small bit that defines how something reacts.
Functional group?
What?
Functional group?
Functional group, right. So remember we had our functional groups like carboxyl, hydroxyl, and can someone name another? Respond. Carbonyl, sulfhydryl, and
It starts with an A. What about amino?
amino.
Oh yeah,
Okay, so you guys got most of them. So that's good. And these define how they interact. So this is, there you go. This is pretty stable, and it won't spontaneously react with most things except for one of these phosphate groups here. So we've got our bases, and let's say that's cytosine, cytosine, yeah. I'm sorry. One easy way to remember is that the base here, cytosine, already has an S in it, so when it binds, it gets a D. It's not the same as that, so it's different. That's the only way I remember.
Yeah. I know that kind of stuff too.
So, overall, this will create a structure where you have a ribose bonded to a phosphate group, bound to a ribose, bound to a phosphate group, bound to another ribose. And so this will have all of your base pairs here. Now, what do these U, C, and G stand for? Uracil, cytosine, and guanine, right? The base pairs that, most of them are still up here. And so when we have ribose with uracil with a phosphate chain, what do we call that? So you have ribose, the nucleic acids, and phosphate groups. Someone said DNA? It's not DNA. It's RNA, because it uses regular ribose. You in the back, you got that. And again, what's the difference between ribose and deoxyribose? It loses an oxygen right here. And one thing I'm going to cover, because it is going to come up later, is that scientists, to be able to say that this specific hydroxide changes, they'll number the carbon atoms. So this one is first. It's most important because it binds to that group of the nitrogenous base. This one's number 2, you're just going around the chain, 3, 4, and 5. So the phosphates will bind at the 5 group and at the 3 group on the bottom. So there's a 3 here, and there's a 5 here that binds to that hydroxide.
This is really important because it's super easy to mess up, and I always mess it up. It's not so like that 5 apostrophe that says it's the 5th carbon, right? That 3 apostrophe says it's the 3rd carbon. And so like pay really close attention to what Jack's about to say.
And that 3 and 5 with the apostrophe just makes it an important number so you don't confuse it with something in the structure. It's just labeling. So now when you have this binding with another segment, I'm just going to draw it over here. It's actually upside down because, remember, this is upside down. So the R is going to want to bind here to the 1 and 1 over there. So you'll have your bases with kind of in the way. Here we go. So this will bind to what? What binds with uracil? Adenine, right? Remember, they have your complementary pairs. I'm hoping some of you wrote this out at your tables because you're going to need to look on it now that I'm like writing over everything. There's not much room on this board. This will go to a phosphate. This will go to another ribose. And what nitrogenous group here will want to match up with cytosine? Guanine, right? And they have the hydrogen bonding there. So it'll be another phosphate. And there's going to be a ribose there. And what matches with guanine? Cytosine, right. Remember, this has uracil because it's RNA. You're using regular ribose. If it didn't have that, what would it be? If it was deoxyribose? DNA. Speak loudly.
Don't be shy. We're all wrong a lot of times, guys.
Yeah, like we learn by making mistakes. So if you shout it out and you're wrong, you know.
Like that's me half the time in class.
Yeah, that's how we learn.
Deoxyribose.
So I heard a lot of people mumble, but no one actually said anything. Right. So if this was DNA with deoxyribose here, because of the structure, instead of uracil, you would have thymine, and it would be a T there. But no Mr. T here. We pity the fool who draws a Mr. T in RNA. And then you're going to have the 5 end here, that's the top, and 3 end there, the 5 end there, 3 there, 5.